So here we're dealing with the problem where we have lizards, we have both male and female lizards, and they all had hatchlings. But what we did was we collected the males and we collected their hatchlings. Now the question that they ask us is, how do we know which hatchlings belong to which father? So in order to do this, we need to do many processes to determine paternity and basically do DNA analysis, perform that in order to determine paternity. So what we first start off with is we need to extract mRNA from collected tissue samples of all the fathers. So we collect them, let's say there's 3 potential fathers. They don't tell us exactly how many fathers there are, but we can just assume there's 3. We're going to collect tissue samples from all of them, and then we're going to extract mRNA. As we know, mRNA is single-stranded and is synthesized from DNA. So now we have that single-stranded mRNA. Now what we're going to do is, what we want is DNA. We don't want mRNA, but we need mRNA to get to DNA. So what we're going to do is we're going to get a sample here. We're going to have a buffer solution. And so what we're going to do is we're going to put this mRNA in the buffer solution. And we're also going to put the enzyme reverse transcriptase, which basically does what its name says. It does the opposite of transcription. So it'll make a single-stranded molecule of DNA. So we're going to also put that inside the buffer solution, and now we have mRNA and reverse transcriptase. So what we're going to do is a process to get double-stranded DNA. That's our end goal in this process. So what we're going to do is, now that a single strand of DNA has been synthesized from reverse transcriptase, and in here is just a bunch of buffer solution, because we can't have it in just dry conditions. So what we're going to do is, now that we have, this is our single strand of DNA, what we're going to do is we're going to add a primer, which is just a section of DNA, and it basically tells DNA polymerase where to bind onto in order to make complementary DNA. Well, complementary DNA to this, which is cDNA. So when we have this, we're going to add a primer, and then we're going to add DNA polymerase. So let's just represent that with blue. So DNA polymerase is going to be here, and what it's going to do, it's going to get the other blue, we're just going to say that it's free nucleotides. So what it's going to do, it's going to bring over those free nucleotides along with DNA ligase. So DNA ligase will also be in here. Let's make it red. And so together they'll work to make a strand of DNA. There. So now technically we have a double-stranded DNA molecule. So then together they make that double-stranded DNA molecule, which is what we originally wanted. Now, within this DNA molecule, there's a bunch of genes that we don't exactly care for, or that we're going to use within the DNA analysis process. So what we need to do is we need to use PCR first in order to get many DNA molecules, because this DNA molecule is very small, and we need much more to do DNA analysis with. So what we're going to do, we're going to get a separate buffer solution to perform PCR with. And PCR is its own little complex process. So polymerase chain reaction, that's what PCR stands for. So what we're gonna do is we're gonna have the 2 strands, and they're not usually separated, but I'll explain why I drew them separated there. The first process is denaturation. So basically we heat up the solution, and so instead of intertwining, they become separated because we want to separate the DNA strands. And then after that, we have to cool it. So this is called the annealing process. So once it's cooled, we have to add a similar primer to what we added here. So we're gonna add a primer. And each DNA molecule will have its own primer. This is on a buffer solution, so all we have to do is add the primer. So once it's cooled, so we're just gonna remove this little fire. Once it's cool, we're going to go into the elongation or the extension process, where we have to add TAQ polymerase, which is just a special type of DNA polymerase. So we'll represent that with green. And that's just going to bring in all the free nucleotides together to make complementary strands to each of them. So I'm just going to draw outside. Basically, the end result of this is that we're going to have two DNA strands, each with their complementary strand. Now we have 2 DNA molecules. So you can see how we went from having 1 to 2 DNA molecules. So over this process, we're going to repeat it again and again and again until we have almost millions of DNA molecules. So obviously once we do this once more, we're gonna separate each one. So we'll have 4 separate, and then you can see that each one will have its own. But now we have 1, 2, 3, and 4. So we keep on repeating this process, and we're gonna get a lot of DNA molecules. So once we have a lot, remember how I said that these genes, not all of them are exactly useful, or we're gonna use in DNA analysis. So what we're gonna have to do is we're gonna have to use restriction enzymes that I wrote up here, restriction enzymes, which basically they just have sites on DNA where they'll cut through the sugar phosphate backbone and they'll create sticky ends. So they're gonna cut some of these, let's say here and here, to create sticky ends. But these restriction enzymes, we specifically chose them so that they can target VNTR regions, which are basically variable number tandem repeat sections of DNA, which this is significant because we need to use those to identify paternity. This is because this VNTR regions are very specific to the organism or the individual, so we can tell differences between species or even the same organism. So once we have, we're gonna put these in another solution. So we're gonna have another solution, and we're gonna put all these DNA molecules here. So what we're gonna do is we're gonna add the restriction enzymes. So the restriction, let's just say that's the restriction enzyme. We're gonna add the restriction enzyme, and we're gonna get a bunch of sticky ends. So sticky ends usually look like this, where there's still regions where nucleotides are attached to one another, but we have free ends, and they're called sticky because nucleotides will want to stick on there with the help of other enzymes. So once we have the sticky regions, or the sticky ends, we're gonna use probes, radioactively labeled probes, to attach to these sticky ends. So we're gonna have these radioactively labeled probes. So now what we're gonna have is basically DNA molecules that look like this. So now we know that this specific DNA molecule has the VNTR regions that we want. So once we have a few of these, there's going to be many of them, we're going to use gel electrophoresis, where we're going to have basically a stage, and we're going to have a negative and a positive side, and so cathode and anode. And so we're going to put these down. First we have to make a gel, so we're going to use agarose gel because this is DNA, so it's just a bunch of gel here. And then we're going to make small little wells within the gel. So we're gonna make small little wells, and then we're gonna put this in these small little wells. We're gonna add glycerine in order to weigh the DNA molecules down. We're gonna add glycerine to do the phosphate bond.